Hi, everyone. Thanks for joining. We're just going to wait a minute to let everyone kind of trickle in. It takes some time to get everyone accepted into the webinar. Okay. All right. So thank you, everyone, for joining today for our Western Blot workshop on lysate preparation. My name is Afrida Rahman-Enyart. I am a scientific liaison and product manager here at Proteintech. Before we begin the webinar, I just want to share a few slides so that I can introduce our company to you. So Proteintech is a leading manufacturer of antibodies, proteins, and immunoassays across several research areas. Um, our portfolio comprises of 3 core product lines. So we have our Proteintech branded antibodies and immunoassays. So we have a very expansive portfolio of antibodies. We also offer antibody labeling kits, ELISA kits, and IHC kits. We also have our humankind branded bioactive proteins. So these are our cytokines and our growth factors. And all our proteins are expressed in human HEC293 cells, meaning that they have native folding, post-translational modifications, and glycosylation. And finally, we also have our Chromotech branded reagents that are based on alpaca nanobodies. And our nanobody-based reagents are ideal for immunoprecipitation, immunofluorescence, and protein purification workflows. So the product lines all share common values under the main Proteintech brand. So we have high quality and validation of the products, and we strive for scientific excellence. But each product line addresses its customers in a separate and specialized manner. I also just want to take a moment to celebrate the success of our customers because our customers' success is also our success. And we're really proud that Proteintech products have been cited in over 180,000 publications worldwide. And we're also really proud that we were the winners of the Researcher's Choice Award last year from CiteAb. So what makes Proteintech different from other companies is that we manufacture all our products in-house and sell directly to the customer. We're also very transparent about our product performance, so we include original validation data as well. So with that, I want to go ahead and begin the webinar. So this is just an overview of what we'll be talking about today. So I want to begin by talking about just a general Western blot workflow, and then dive into more of, what goes into Lysis solution for your lysate preparation, how to lyse samples effectively, so, cells versus tissue, and really the value of sonication, whether or not a sample always needs to be boiled, and then go through some lysate troubleshooting. So at the very end, we'll have a Q&A session. So I'll be joined by Dr. Rick Liu, who is part of our tech support. So he'll be helping with answering some of the questions that you guys have at the end. So Western blotting is a technique that allows you to separate and detect proteins of interest from a sample. So you can identify your protein of interest from a complex mixture of proteins, such as from a cell lysate. So the first step before beginning your Western blot is going to be to prepare your lysate. So this is going to be the main topic of today, but just very generally, people commonly use a RIPA buffer or some chemical-based lysis buffer that includes a detergent in order to disrupt cell membranes and release proteins from cell or tissue samples. After that, a loading buffer that contains, SDS, beta-mercaptoethanol, bromophenol blue, and glycerol is generally added to denature and uniformly coat your proteins with a negative charge. So once your sample is prepared, it is ready to be loaded on an SDS polyacrylamide gel. And so the gel is placed between two electrodes in a chamber with running buffer that contains SDS, one with a negative charge here on top and one with a positive charge at the bottom. So an electric current is then applied, and since all of the proteins in your sample are coated with a negative charge, uh, they begin moving down through the gel towards the positive charge. So the gel is like a grid or a mesh. So you'll begin to see that smaller proteins move through the gel more quickly than the larger proteins. And so this process allows the proteins to be separated on the gel by size. The proteins on the gels are then transferred by electroelution onto a PVDF or a nitrocellulose membrane to allow antibody detection on a surface that's more durable. So these types of membranes tend to have a high affinity to proteins, including antibodies. So it's really important that you minimize nonspecific binding to the membrane by doing a blocking step. So after blocking your membranes, you can then incubate with primary antibody that's specific to your protein of interest, followed by wash steps in order to remove any unbound primary. And then you can incubate with an HRP or fluorescently labeled secondary antibody, which recognizes your primary antibody. This is then followed by another wash step to remove any unbound secondary. And then the last step is going to be your detection. So if you're using an HRP labeled secondary, your membranes will be incubated with a chemiluminescent HRP substrate prior to imaging. And if you're using a fluorescently labeled secondary, you can use a digital imager. So that's a lot of steps. So of course, with so many steps and chemical interactions, something is bound to go wrong, and Western blotting can be prone to having some troubleshooting issues. So some common troubleshooting issues that we have seen are having, too many bands or having bands that are streaky or misshapen or having ladder-like staining. So from the experiences of our tech support and our R&D, we've seen that these types of problems generally arise from issues with lysate preparation and storage. So, a wise scientist once said that a successful Western blot relies upon 10% reagents, 10% execution, 10% luck, and 70% protein extraction. Of course, these percentages change from scientist to scientist, but when our team at Proteintech validates antibodies for Western blotting, we make sure that we test multiple sample types for each antibody in order to optimize protein extraction. So in our experience, of helping customers troubleshoot their experiments, we've often seen that sample preparation is really the key area where most improvements can be made in order to ensure for clean blots and making sure that you have reliable and reproducible results. So the preparation of cell lysate is crucial to the success of many assays, including techniques that use antibodies. And ideally, throughout the whole process of sample preparation, from the time you collect your lysate to analysis, your protein of interest should be well preserved and protected from the activities of things like proteases and phosphatases. Also, proper storage and handling of the lysate are crucial in order to avoid any degradation and maintaining the ability to detect your protein. So what is the goal of lysate prep? So the main aim when you're doing your lysate preparation is to solubilize your proteins by breaking down protein interactions. So this is things like your disulfide bonds, hydrogen bonds, van der Waals forces, ionic interactions, and hydrophobic interactions. And ultimately, by doing this, you get down to the linear form of your proteins, and this avoids protein aggregation, precipitation, and sample loss. So what exactly goes into a lysis solution? So there's really four main components to consider, and we'll go into a little to each of these more individually. So you want to consider your buffer system, adding chaotropic agents and detergents, protease and phosphatase inhibitors, and the addition of reducing agents. So when considering your buffer system, it's really important to make sure that you maintain the physiological pH of the solution. So if the pH is out of physiological range, this can lead to protein precipitation, instability, and the formation of insoluble products. So in order to avoid this, you want a buffer system like Tris HCl, which is most commonly used. Um, there are other buffer options, such as you can use a HEPES buffering system. But one thing that we do recommend is to avoid buffers with high concentrations of potassium, because when you mix potassium with SDS, this can lead to the precipitation of proteins. Additionally, you want to consider salt ions in your buffer to maintain ionic strength of the solution for the proper disruption of cell membranes and cellular interactions. So again, be careful when deciding on salt concentrations though, because high salt concentrations can actually damage your proteins of interest and cause precipitation. So if you have too much salt present, you might begin to notice arch-shaped bands on your gel. So you want to make sure that you keep your salt concentration between about 50 to 150 millimolar. Something else to consider in your lysis buffer is the usage of chaotropic agents. So chaotropic agents weaken the hydrophobicity of proteins in order to make them more soluble in solution. So some common chaotropic agents are urea or thiourea and detergents. So urea and thiourea work by disrupting hydrogen bonds between amino acids, and this results in the unraveling of the hydrophobic regions. So in lysis buffer, it's recommended, if you're going to use urea or thiourea, to use about 6 to 8 molar urea or 2 molar thiourea. Another option that you have for chaotropic agents is at the addition of detergents. So detergents are really interesting because they have both hydrophilic and hydrophobic properties. So the way that detergents work is that the hydrophobic ends will bind to the hydrophobic portions of the proteins in the lysate, while the hydrophilic ends are going to interact with the water. And this then results in the solubilization of your proteins. So this chart here shows some common detergents that are used in lysis buffer. So you can range from strong ionic detergents to non-ionic detergents. And the usage of detergent is probably one of the most complicated parts of making your lysis buffer. So it's really quite challenging to predict the optimal concentration that's needed to extract every single protein. So what we recommend is experimenting with different types of surfactants, especially if you're looking at membrane proteins, because those can tend to be a little bit more tricky. So if there are so many additives that you have to consider, how do you know which lysis buffer you should use? So the first place that you can begin is by considering the cellular location of your protein of interest. So if your protein of interest is soluble and cytoplasmic, you might want to try a Tris HCl buffer. If it's cytoplasmic and cytoskeletal bound, you could try a Tris Triton buffer. If you're looking at whole cell lysate or a membrane bound protein, you can try an NP40 buffer. And then if you're looking at a nuclear, mitochondrial, or membrane-bound protein, you can consider a RIPA buffer. So at Proteintech, we always use RIPA buffer for our Westerns, but every protein is different. So you want to make sure that you familiarize yourself with the literature first before you decide on the buffer. So the next thing to consider in your lysis buffer is going to be the importance of using protease and phosphatase inhibitors in your lysis buffer. So all living organisms contain proteolytic enzymes. In whole cells, however, this reactivity of proteases and phosphatases are tightly regulated and compartmentalized. But once your cell is disrupted by lysis buffer, your cellular and tissue architecture is also disturbed. And what this means is that the proteins in your lysate can all potentially be accessed and targeted by proteases for degradation or modification. So therefore, protease inhibitors, in order to inactivate or block activity of these endogenous proteases, are really critical for preserving your target protein. So there are several classes of protease inhibitors that are available, which each target different types of proteases. So the most commonly used and highly effective inhibitor is PMSF, which targets serine proteases. Other inhibitors include E64, which targets cysteine proteases, pepstatin A for aspartic acid proteases, and EDTA for metalloproteases. So many protease inhibitors also require a divalent metal ion in order to function. So oftentimes a sequestering agent like EDTA is recommended. So we recommend that you use a protease inhibitor cocktail so that you make sure that you're actually targeting all these proteases. So you can see here on this blot on the right, just how different your results can look if you're using a protease inhibitor cocktail versus if you're not. So in the lane without the inhibitor, you can see that we have more bands signifying increased protein degradation compared to when we do use an inhibitor cocktail. So if your protein of interest is a phosphorylated protein, then the other thing that you might want to consider is also using a phosphatase inhibitor, such as sodium fluoride or sodium orthovanadate, so that you can preserve the phosphorylated form of your protein as well. So prior to loading samples on a gel, an SDS sample buffer is usually added to your lysate. So sample buffer generally contains SDS, glycerol, bromophenol blue, and reducing agents such as beta-mercaptoethanol or DTT. So as I mentioned earlier in the presentation, the SDS and the reducing agents are required for the coating of proteins with a negative charge and the denaturation of the proteins. The bromophenol blue and the glycerol are added for the ease of loading of samples, since the dye allows you to see the samples and the glycerol makes the samples more viscous. So I want to highlight the importance of using reducing agents in your SDS sample buffer. So many proteins exist in multimers through disulfide bonds. Reducing agents disrupt these bonds by donating electrons so that the extracted proteins are present in their monomeric form. So if no or insufficient amount of reducing agent is added, this will result in an incomplete reduction of the protein and the presence of dimers and polymers. So you can see an example of that on the blot here on the right. So on the left side, we used 5% BME, and on the right, we used no BME. And you can see that we have BMP4 multimers in the condition with no BME. And it's also important to note that if your samples are stored for 6 or more months at -20 or -80 degrees Celsius, then you need to add more reducing agent in order, more reducing agent, and the samples should be reboiled. Sorry, guys. So after preparing your lysis buffer and making sure it is ice cold, you are ready to lyse your samples. So effective sample lysis and homogenization is key to ensuring that cellular structures are broken down and proteins are released into the solution so that they can be accessed. So for cell cultures, lysing samples is quite straightforward. So you first want to make sure that you pre-chill your centrifuge to 4 degrees and that you have your reagents on ice before starting. If your cells are in suspension, you want to make sure that you centrifuge them so that you pellet the cells. And after washing the pellet with PBS, you can then use something like a RIPA buffer. And we recommend using about 100 microliters per million cells. If, on the other hand, you're working with adherent cells, you want to make sure that you first remove the culture medium and then wash your cells with PBS. And then you can add RIPA buffer either directly to the well and lyse in the well, or you can scrape and collect these cells into a tube. So you want to make sure that you avoid using trypsin in order to release your cells, because trypsin proteolysis will actually degrade your proteins. Um, but I know that that's not always something that you can do. So if you absolutely need to use trypsin for, like the more pesky cells that you have, then you want to ensure that you wash your cells thoroughly prior to lysing. So on the right here is an example of a recombinant SARS-CoV-2 protein with and without the incubation with trypsin. And you can see that when trypsin is added, we see many bands indicating that we have protein degradation here. You can also prepare samples for Westerns directly from tissue. So the key to good sample preparation when using tissue is making sure that you have a clean dissection. So you want to make sure that you wash the tissue in cold PBS to remove any contaminating blood products that you might have. So blood products contain endogenous IgG, and these endogenous IgGs can actually bind non-specifically to your secondary antibodies, and they're going to recognize, your secondary antibody is going to recognize the heavy and light IgG chains. So an example of this is shown here on this figure on the right, where we see mouse spleen and thymus tissue for Western blot detection of actin. And we used an anti-mouse secondary antibody here. And you can see that we, in addition to seeing our actin band, also see contamination of the heavy and light IgG chains on the blot. Other things that you want to keep in mind when you're preparing samples from tissue is you want to make sure that you do everything on ice, even if you're just mincing the tissue. Also ensure that your tissue is completely homogenized, and this is going to vary based on your sample type. So for example, if you have higher connective tissue content, then you might need more cycles of homogenization. So in general, we recommend using about 500 microliters of RIPA buffer per 10 mg of tissue. Something else to consider for lysate preparation is the value of sonication. So at Proteintech, we always add a sonication step to our lysate preparation for both cell and tissue lysates. This step helps to further break up the cells or the tissues to fully solubilize the proteins. So sonication also helps to overcome issues with, high lysate viscosity, which presents issues with sample handling and pipetting. And it can be seen as, streaky sample lanes on a gel, as you can see here on these examples. So high viscosity typically arises from a high nucleic acid content in samples because DNA molecules are very long and they're very sticky. So sonication allows for the shearing of the DNA, and this will, in the end, reduce your sample viscosity. So sonication is recommended over the use of things like DNase because DNase can actually introduce other protein contamination to your lysate. It's also important that you don't oversonicate the samples. So you want to make sure that you perform sonication cycles at 10 seconds on and off for 1 minute for cell lysates or 2 to 5 minutes for tissue lysates. And you want to make sure that you keep your samples on ice because one of the issues with sonication might be overheating. So you want to just make sure that everything is on ice and that you do these sonication cycles. So the next part of lysate preparation, is getting your samples ready for loading. So you want to determine, the amount of protein that you have and how much protein you want to load onto your gels. So you might actually need to dilute your samples first. After that, you'll add, an SDS sample buffer, and generally lysates are boiled after you add your SDS sample buffer. However, the question really is boiling always going to be the best option? So typically, SDS sample buffer is added to lysates and it's boiled for 5 to 10 minutes. Heating the sample allows for the disruption of hydrogen bonds, hydrophobic interactions, and van der Waals forces, which allows for the unfolding of the proteins. And this process, after you heat your sample, is irreversible. One common issue that we have seen with boiling is that membrane-bound proteins can often aggregate after you boil your sample. So for example, here on the right, we are looking at the sodium potassium chloride transporter SLC12A1. And when samples are boiled, it is not possible to detect the protein on the membrane in the Western blot. So the reason for this is because if you have this protein aggregation, then your protein complex is the molecular weight is too high. So the protein isn't going to run through the gel or transfer. So what's the solution in order to prevent protein aggregation? So one thing that you can do is try lowering the heat from the traditional 95 degrees Celsius. So you can see here on the right just how different results can be if you just reduce the heat. So here we used mouse brain lysates that were heated at various temperatures, and you can see that between 4 to 75 degrees, you are able to detect the protein of interest. However, once the lysate is heated to 85 degrees and beyond, our protein begins to aggregate and become trapped at the top of the gel. Another option to prevent protein aggregation is through the addition of chaotropic agents such as urea to help disrupt hydrogen bonds and increase the solubilization of your proteins. So for the last part of this webinar, I want to go over some common troubleshooting issues for lysate preparation. So if you are seeing diffuse staining of your gel lanes with wide bands, similar to the ones we're showing right here, this is a telltale sign of high lipid content in the lysate. So like nucleic acids, lipids can bind to proteins and cause issues with your Western blot by blocking the gel pores and affecting separation of proteins in the gel. So when working with a lysate from fatty tissue, such as adipose tissue, you want to make sure that your lipids are properly removed during the dissection step. So if there are still some fatty tissue that remain after dissection, what you want to do is you want to make sure that you incubate samples on ice or in the fridge for a few hours in order to precipitate lipids. And this will help with the high lipid content. If you see gaps at the top of your gel, similar to the gels right here, this indicates that the protein can't migrate through the gel. And this can occur for several different reasons related to your sample prep. So the most common reason is going to be that you have a high lysate concentration. So when your lysate concentration is high, during the denaturation step, the proteins can actually aggregate into very large complexes and they can't migrate. So when this happens, the protein complexes are completely denatured and they cannot be disaggregated. And because the denaturation step is irreversible, we recommend that you actually return to your original lysate, dilute it, and then carry out the denaturation step again. So in order to avoid this happening, in the first place, we do recommend that you run a protein assay first so that you can determine exactly how much protein is in your sample. Another reason for a gap at the top of your gel is that the samples have been stored for too long and your reducing agents have oxidized or they have evaporated. So you're going to see that protein integrity is going to be at its highest in freshly prepared lysates. And so in fresh lysates, you'll see a low level of protein degradation and protein complex formation. But as lysates are stored for a long period of time or the number of freeze-thaw cycles increases, the reducing agent components in your sample, for example, your DTT or your beta-mercaptoethanol, they're going to gradually oxidize and become invalid. Due to this, the denatured state will become unstable and you'll see protein-protein complexes beginning to form. And then when the lysate is then run on an SDS-PAGE gel again, the protein complexes can clog the pores of the gel as shown here on this image. The way that you can fix this is by re-adding more reducing agent and applying the heat denaturation step again to disaggregate any protein complexes. So for example, here we have re-added DTT, and you can see that now we don't see the protein complexes, and that the proteins are able to move through the gel. So sample degradation is also something that I want to talk about because it's the first and most likely thing to happen to a stored lysate. So boiling of samples initially results in, the denaturation and the inactivation of fundamental proteins. And this is going to inhibit the degradation rate of the sample. But as the storage time increases, the degradation of your sample will gradually be amplified as well. So the figure here on the right shows comparison SDS-PAGE gels for protein samples, which have been run just after lysate preparation and after several years in storage. So following long-term storage, there's a reduction in the number of protein bands present, which appear blurred, and particularly here in the upper part of the gel. So we do recommend that if you're planning on storing samples for a long period of time, to do so in -80, and if you store it in -20, not to do so for longer than 3 months. So overall, the take-home messages of this lysate preparation workshop is to make sure that you choose an appropriate lysis buffer for your protein of interest and include protease and possibly phosphatase inhibitors based off of your protein of interest. You want to make sure that you use fresh lysates where possible, and also remember the value of sonication. And lastly, if you use lysates after long-term storage, make sure to check your protein complexes by SDS-PAGE. So with that, I just want to share a couple more slides before we begin the Q&A session. So I just want to share a couple product slides that might be interesting for our audience today. So in addition of Proteintech having antibodies validated for Western blot, we also offer accessory products for Western blotting, including loading control antibodies. So we have antibodies for various molecular weights and protein localization, and we also have the highest cited GAP-DH antibody. We also offer protein ladders and our Signal Bright ECL substrate. And our ECL substrate can detect proteins at femtogram levels, which makes it ideal for your low abundant targets and your precious samples. And lastly, I just want to talk about our flexible antibody labeling kits. So flexible is a novel antibody labeling kit that uses an affinity linker in order to conjugate fluorochromes, enzymes, and molecules in any buffer condition in just 10 minutes. So this means that you can label your antibody for fluorescent Western blotting and eliminate the secondary incubation step. Because you label the antibodies prior to incubation with your samples, flexible is suitable for same species multiplexing. So you don't have to worry anymore if your favorite antibodies are all from the same species. You can use those antibodies for your fluorescent Western blot experiments. If you enjoyed today's webinar and you want to learn more, you can check out our video hub on our website. We have other workshops for other applications such as IHC, chip and IP workshops. Also, if you want to keep up to date on our future webinars, please do follow us on our socials because we post about our events there. And then also as an attendee of today's webinar, you will receive a follow-up email that will have a link to request a free 20 microliter antibody sample. You will also receive a certificate of attendance and also a recording of this webinar. So with that, I'd just like to thank everyone for attending the workshop. If after the Q&A session you do have any questions, please don't hesitate to email us at our events email at events@ptglab.com. Or if you have any technical questions, you can email our tech support at tech-us@ptglab.com, or you can email me directly at afrida@ptglab.com. So I've noticed that we have some questions already, but if you do have any questions, please do drop them into the Q&A box. And I will go ahead and begin the Q&A session and welcome Dr. Rick Liu, who will be helping me out here. He is a part of our tech support team. So let's see. There's a lot going on. Okay. Yes. So you guys will receive a recording of this talk, and I can also include the slides as well. Okay. So such a friendly group. Everyone says hello. Trying to so Nicole's asking about phosphorylated proteins. So the main inhibitor that I would recommend is a sodium orthovanadate. Dr. Liu, I don't know if you had anything that you wanted to add with inhibitors for.
That was my answer too.
Awesome.
I mean, for the phosphatase.
Yes.
Yeah. And it's very cheap too. So.
Yes. And that's always good.
It won't hurt. It only helps.
Did you say that homogenates and sample buffers should be boiled again if they are stored in -80? So we recommend that if you need to store your samples at -80, not to add your sample buffer yet. So you want to take your lysate and then add the sample buffer and boil. That would be ideal. However, if you do have your sample buffer already added, you can try boiling, but you also don't want to overboil.
Yeah, that really is important because in SDS sample buffer, actually the proteins are not as stable. I mean, against proteases.
Right.
They are actually more prone to be degraded somehow.
Okay. So will water bath sonication work, and how long is necessary? Dr. Liu, do you mind taking this one?
Probably not.
Okay.
Yeah, and the water bath sonication is just not powerful enough, I guess. I don't see much help with the water bath sonication. So I have to use the probes that goes into the tube.
Yeah.
And also I want to add that sonication is definitely good. It helps. And some people, what I want to say is you cannot use sonication to substitute for, the glass homogenizer, especially for tissues. I think sonication works well for cell cultures, for the cells suspension. But for tissues, you really have to do at least two things. You have to homogenize really well, and then you can sonicate. So, but there are people, they just sonicate the tissue directly, but I don't think that's enough. It doesn't really substitute for homogenization. So,
yeah. Someone's asking for any additional troubleshooting recommendations for cell membrane protein preparation other than lowering temperature. I think in this situation, you would really need to mess with, the different types of chaotropic agents you use in your lysis buffer, what kind of detergents you're using. I've also found that, vortexing has helped with membrane bound proteins. Dr. Liu, did you want to add anything to that? Do you have any other tips and tricks for membrane bound proteins?
well, basically, yeah, detergent is the key. So if you can get away with just tritone, non-ionic detergent to solubilize the membrane protein, that'd be great. But always, I guess adding a little bit SDS helps. So like a Proteintech, we also always use wrapper buffer, which contains some SDS and of course MP40. So that combination works the best for membrane proteins, really.
Thank you. So someone's asking, do you perform Western blot on cell culture media for released protein detection? I have personally done this before. It depends on what you are trying to detect. One of the issues is going to be that you won't have a lot of your protein of interest in your culture media. So that might be one of the difficulties that you face. So you might need to use, a more sensitive detection substrate. But I don't know, Dr. Liu, do you have any other?
If you can concentrate, that'd be nice.
Yeah, exactly.
Maybe concentrate a little bit, maybe precipitate the protein of interest from the media. Yeah, I totally agree with you. Basically, the key is really to how to increase the level of the protein for the detection. So
So someone's asking if we recommend boiling before storing a sample at -20. So for Westerns, your sample is going to be best if you use it fresh. So obviously, if you have your sample at -20, you would want to thaw it and then boil it with your SDS sample buffer. So I would recommend boiling it after.
yeah, that should work. But really, the best is to always use it fresh.
Right.
I try now to store it even overnight.
I know that's difficult. It's so difficult to do.
If you have to, yeah.
So what is the best method for homogenizing tissue? Can we use RIPA buffer for preparing total brain extract? So that's obviously going to depend on the type of tissue. So here you're using brain extract. So brain is pretty easy to homogenize. So I've actually used like a 1 ml glass pipette and been able to use RIPA buffer to just homogenize tissue directly. So that would be my suggestion, is to, if you don't have to use something too aggressive, don't. So just a 1 ml glass pipette and just pipetting it up and down for brain extract works.
Yeah, you can always homogenize if you want to. I mean, it never hurts. You basically want to get the best, you want to solubilize everything. So
so this is a good question for you, Dr. Liu. How important is flash freezing original lysates in liquid N2 or on dry ice versus sticking the samples in the -80 straight away?
Well,
'Cause
the flash, the quick freezing is good, always probably better. But my guess is maybe in practice, it's not much different. You probably will not see any significant differences. But if you have the the setup, you can do a quick freezing. I think that may be better. But I have not noticed any in my practice. I have not seen that much significant difference. So, but I probably psychologically I will feel better if you do the quick freezing, the flash freezing. So,
so Dr. Liu, if you know a little bit about how we sonicate our samples, So someone's asking if we can talk a little bit about how we sonicate the samples. How much sample at a time do we sonicate?
I try to, for Western blots, I try to aim for about 10 milligrams per ml concentration. So if you kind of have idea, what the size of your tissue, so you have. So let's say if you start with like a 10 centimeter plate of cells, confluent cells, and you can probably do like a half ml of volume of, let's say, a wrapper buffer, then you can sonicate that. And usually the time will be 30 seconds to a minute on ice, because it heats up really quickly with those sonication probes. So keep it on ice, and if you have to stop and pause, maybe a few seconds. But a total of 30 seconds sonication is usually enough.
Yeah, you want to, that is the one difficulty with sonication, is just making sure that you do not overheat your samples. So someone is asking, how do you remove fat contamination from lysate effectively if it's not the top layer? So one thing that you can do is you can put it on ice, or you can put it in the refrigerator for a little bit and allow the lipids to kind of precipitate. And once they precipitate out, it'll be easier for you to remove the fat contamination from the lysate. There are so many good questions. So someone's asking if you can store cells in -80 and then prepare the lysate later. If yes, how long is better? So I guess how long can you store it for? Do you know the answer to this, Dr. Liu? If you can store cells?
I have some thoughts. Yeah,
okay.
I prefer, actually, that's a good idea. I prefer to store the intact cells in -80 instead of nice cells, because when you nice the cells, you basically disrupt everything. You release the proteases, everything is messed up. So it's better to keep the cells, without lysis in -80. So that's actually a better way, I think. The cells will be still dead.
Yeah.
but at least everything will be still in place, and there'll be less protease activities in that way.
Yeah. So someone's asking, what is the best and fastest method for measuring protein concentration? So there are companies out there that sell these types of assays. So they're pretty quick to do. They take about like 15, 30 minutes to do the assay, and then it's like a colorimetric type of result. So I know there are companies out there that sell these kits so that you can determine your protein levels.
Yeah, I would add, of course, you can always do a absorption if you have a spectrometer. Do a 280, 260 absorbance. I used to always keep a little bit Bradford solution on the bench. So the Bradford assay is very easy. Basically, it's a dye reaction. So most of the times you can just take maybe 10 microliters of your lysate, and the lysate, you just add the Bradford solution. Then you can see the color change and maybe do a absorbance. That's pretty quick and easy. So
yeah. Someone's asking for any advice for the extraction and detection of extracellular matrix proteins from tissues, i.e. collagen. So generally, we recommend using a RIPA buffer. Of course, it's going to be different based off of whatever your protein of interest is. So you might need to try several different types of buffers, but in our hands, RIPA buffer tends to work well with most proteins.
Yes, because the SDS, and also I will add that collagen is a little bit tricky or unique because it tends to form bundles on their own, and you need to try different like extraction conditions. I know some people maybe they use acidic solution to solubilize collagen, and they seem to work well. But RipoBuff should work too. So for at least for Western blot.
Right. So someone has a question. They've been trying to detect SLC26A3, a solute carrier, using Western blot, but all they observe is a blob at the top. The proteins aggregate and don't move through the gel. I have tried the no boil method, which hasn't made a difference. I use RIPA for Lysis Prep. So in this situation, you might want to mess around with the chaotropic agents you use. You might try using some urea or adding different amounts of the detergent. Dr. Liu, do you have any advice on what they can do?
Yeah, this kind of proteins, I mean the ion channels and mitochondrial membrane proteins, they can be tricky to actually to solubilize. Yeah, definitely maybe increase the amount of SDS in the Lysis buffer, and also make sure you have fresh DTT in the during the Lysis. So I think a lot of people think your SDS sample buffer has DTT or beta mercaptoethanol, and you will think when you boil the sample, everything should be denatured and basically reduced, right? But sometimes during the lysis process, if the proteins form disulfide bonds between two proteins or maybe intermolecular disulfide bonds, and those disulfide bonds can now be reopened even when you are boiling with the SDS sample buffer. So that could be one of the reasons. So make sure in the Lysis buffer you have a reducing agent. So that would be my suggestion. So.
There's another good question for you, Dr. Liu. Do you recommend Western blot technique methods for secreted proteins such as substance P?
Gosh, that's a tricky one. So basically, when you're dealing with the secreted proteins, from the tissue, let's say from culture cells, you are trying to catch them before they're secreted. So that usually is not different, and basically you can just treat the cells as what you normally do. But if you are dealing with the tissue samples, so I would imagine the secreted proteins will be secreted, most of them. And when you trying to homogenize the tissue, you don't want to lose those secreted proteins. So if you keep everything, all the lysates, you should be fine. So just be careful, I guess, pay attention, and you should get most of the materials.
Thank you. So we have another question asking, when collecting cells from a plate or dish, should we keep those on ice while collecting or just scrape into pre-chilled tubes? So I would recommend that as soon as you're ready to lyse, I would do everything on an ice bucket. So this is, removing your media, removing or adding your PBS, keep everything chilled. Because as soon as you remove your media or you're adding PBS, you have the risk of your proteins degrading. So immediately put that on ice right before you add your lysis buffer.
Yeah, I totally agree. Yeah, well, when you get ready, first get a bucket of ice, get everything on ice. Do your best.
So for a sure result, what's the most valuable step in immunoblotting? Lysate preparation, making sure that you have, I know I mentioned this in the talk, but knowing how much detergent to add into your lysis buffer is probably the most important step in figuring out, how to solubilize your protein of interest. Do you have a different opinion, Dr. Liu, or?
I agree, totally agree with you. I mean, because the lysis process is most of the variations come from the lysis preparation, really. I mean, when you run gel, it's pretty standard, and everybody probably does it differently, but eventually they're all the same. I mean, all the rest of the protocols, they are pretty standard, and most of them are pretty consistent. It's the lysis preparation, there's a lot of variations, and people use different buffers, and they homogenize differently. So that's where most of the issues come from.
So someone's asking if we had any specific advice for Western blotting on adipose tissue regarding sonication or your tip for precipitating lipids. So those would be the main tips, making sure that you, sonicate your lysates, and then also, you want to make sure that you have your clean dissection, but afterwards, if you need to precipitate lipids, to either chill it on ice or to keep it in the fridge for a few hours.
Sounds good. Yeah.
Someone is asking about the free sample antibody for the attendance. So we'll send out a follow-up email that's going to have a link. And the link, when you follow it, it'll have a form, and you just give us the catalog number of the antibody that you want and the mailing address, and we'll have that sent over to you.
Yeah, there's one question about the native gel.
Oh yeah.
Do we follow the same technique? So basically, native gel means basically the proteins are not denatured, right? So the sample buffer does not contain SDS, and so your lysis buffer should not contain SDS. So that's really the only difference.
So I think we have time for one more question. If we didn't get to your question live, if you included your email in the Q&A box, or I have the emails on the chat, we can reach out to you by email and let you guys know the answer to your questions. So I'm sorry that we didn't get through all of them. There's so many, which is great. I'm glad you guys are participating. So someone's asking, how can they remove any polymer contamination? What is the source of polymer contamination? Do you want to take this one, Dr. Liu?
Polymer, I mean, DNA or nucleic acid? I'm not sure the polymer.
I'm assuming they mean like aggregation of proteins.
Oh,
possibly.
I see. So basically, I guess the best way to deal with this is when you at the beginning, the first step when you lyse the material or the tissue or the cells, keep everything like really solubilized. That's the key. To prevent them to form the aggregates in the very beginning. Because once the aggregates are formed, it's very hard to disrupt them or to dissociate them. So basically, the key is in the very first step. When you add the Lysis buffer, make sure the Lysis buffer is the volume is large enough. And it's the right, has the right ingredients, with the salt, detergents, ionic and non-ionic detergents, all those things. So if the buffer is good, and everything should be solubilized.
Great. Well, thank you so much. Thank you everyone for attending today. As I mentioned, if we didn't get to your question live, we will reach out to you by email. And thank you, Dr. Liu, for joining.
Thank you, Freda.
Yeah, if you guys do email tech support, you might be interacting more with Dr. Liu. So
Yeah,
I hope
definitely.
you guys all found the workshop valuable, and please do follow us on our social media for any upcoming events that we will have. So with that, have a good rest of your day, everyone.
Great, thank you.